All right, I give up. Can't get into my box. Closed. I'll load the videos and show you guys tomorrow. I have a mitosis, a meiosis, and then after fertilization, the mitosis going on. So I just want to show you guys those. They're fun. All right, so we're going to start chapter 13, genetics. I posted one little tutorial about sex determination, just the monohybrid cross, and then there's a little practice sheet. I don't know how many practice problems are in the back of this textbook, I can't remember, but I posted some little questions, and then I'll post the key tomorrow, so you guys can go through those and make sure you can do these problems. Okay, so Mendelian genetics is named after Gregor Mendel. Right? He worked out the rules of inheritance using garden peas. So he spent a lot of time in his garden. He did a lot of observations before he started his experiments, and at least in the garden pea, there were, for the traits that he ended up following, they only showed two different phenotypes. So pea plants were either really short or tall, yellow or green, smooth or wrinkled, and then a whole bunch of other traits. But there weren't any medium-sized peas, not like people. People is a bell curve, right? From pretty short to ridiculously, to average to ridiculously tall, right? That's a continuum. Anytime there's a continuum in any trait, it generally means it's not a one-gene trait. Because if it's a one-gene trait and there's essentially two alleles, you're going to get maybe 2 or 3 phenotypes, but they're going to be very distinct, right? You're not going to get sort of that bell-shaped curve. So anything where you get a bell-shaped curve of a trait, height, weight, hair color, eye color, skin color, multiple genes involved in determining what those phenotypes are. When they're very distinct, tall and short, it's generally a one-gene trait, right? Attached or detached, one-gene trait. You have a straight forehead or the widow's peak, you know, little vampire guy, that's a one-gene trait. Tongue rolling or not, you either have the gene or you don't, or the allele. If you can't do it, it's not because you're not trying hard enough. Okay, you literally do not have the musculature to turn your tongue like that. Okay? Hitchhiker's thumb or not, like does your thumb bend backwards or does it go straight? It's a one-gene trait. Okay? So he picked things that were very discrete and distinct because you can't follow something that's on a continuum because you get too many variables. Right? As scientists, you try to eliminate all the variables to come up with some sort of hypothesis about what's going on. So he chose traits that were very distinct, and then he watched how they were passed. 2 shorts, do any talls ever come up? 2 talls, do any shorts ever come up? Over and over and over for all these different traits. Trying to figure out how genetics are passed down. Right? Sometimes offspring look like their parents, and sometimes they don't. Right? That's where it all began, but using a simple system instead of a ridiculously complex system to study this. And then if you have a bunch of people from one plant, it may have, 20 to 30 of those pods, which each one contains 10 or 15 seeds, lots of offspring to look at, right? If you use mice as your model system, they might have 12 offspring, and it takes, 3 months to get those 12 from a pea plant. You might end up with, 500 to 1,000 from one crop. The numbers matter because all of this, all of genetics essentially is probability, right? Which chromosome got into the gamete that made that offspring? It's probability, right? What are the chances that grandma went this way and grandpa went that way and there wasn't any grandpa on grandma that went that way? So it's all probability, and probability and numbers for probability only matter when there's lots of numbers, right? I bet all of us could throw 10 heads in a row on a quarter, right? That's totally possible. Probability says it should be 5 and 5 because there's 2 sides to the coin, right? It's a 50-50 chance every time you throw it. But we know you can get 10 in a row, right? Does that mean probability is stupid and we shouldn't ever learn anything about it? maybe. No, it just means the numbers are too small for probability to work, right? They never claimed probability is what happens. It's the likelihood by the numbers game of what should happen. Is it likely that you'll throw heads 100 times in a row? No. Really, really unlikely. So 100 is a decent number for 1,000, what about 1,000? No, it's not going to freaking happen. So that's an even better number for probability. Okay? So anytime we're talking about small numbers, it's always the likelihood or it should happen that way. Doesn't mean it will. Okay? So he used these because it gave him lots of numbers and he could play out in the fields and look at all kinds of stuff. So at the time, in the 1800s, the blending inheritance was what people thought happened. Nobody had any idea about genes or chromosomes really, or how meiosis worked or anything. Right? They were just guessing. And so sometimes offspring look like a blend of their parents. So somehow they get all mixed up. And so, oh, but look, when we have a black sheep and a white sheep, we get a gray sheep. Right? Sometimes you do. Sometimes you get black sheep, sometimes you get white sheep. Right? But there are certain traits that do appear to blend, and so they said that's how it works. But Mendel said that that's crap because of all the stuff he saw with his little peas, and that sometimes a black and a white sheep, you get black sheep. So that didn't blend. So the rule of blending could not be universal. So he wanted to figure out how all these traits were passed. And again, model organisms that we use should have a short, well, lifespan doesn't matter so much, but generally a shorter lifespan means an even shorter gestation period, so you can, get lots, you get several rounds of crosses of peas all summer long, right? Should be inexpensive to keep. You get lots of large numbers of offspring, again, that's probability, and something easy to work with. So some really common model organisms that we use in the lab now are, I'm drawing with white, that's helpful. Yeast, a little roundworm guy, a mouse, and Drosophila. And so mice are not ideal systems for genetics because you don't get a ton of offspring, but they're a great system because they're mammals, and as it turns out, they're really similar to us in a lot of genetic ways, a lot of metabolic ways, and their immune system is really, really similar to humans. So lots of studies involving immune systems and drugs and cancer treatments are amazing to work with in mice because they're very similar to how their physiology reacts to things like ours does too. And so we wouldn't use mice to do our first experiments on genetics or first experiments with drugs, right? But they are a really good model system to go to before we go to clinical trials to make sure everything's safe and it looks like it's going to do something good. Okay. The features of our peas, like I said before, plants have both male and female parts on the flower, right? And so they can do themselves if they want to, or we can cross-pollinate. And he could manipulate this by artificially making a flower either male or female. So, he's out there in his field with his little scissors, and he's either cutting off the male parts, everybody cringes, or cutting off the female parts and essentially making one whole plant female and one whole plant male, so they can't self-fertilize. So he can see who's the mom and who's the dad, what their phenotypes are, so he can follow stuff, right? And then you can also do a self-fertilization if you just want to see what happens, that one plant on itself. Yeah. No. If you whack it, sorry, if you whack it off, I should use that terminology. If you cut the parts off, sorry, it's an accident, then they don't regenerate. Yeah, so and a lot of times if you get, there's some of the lily flowers that you get in bouquets, they cut the male parts off because the pollen is, it's a really big, thick, full of pollen, and it makes a mess all over, and people are really allergic to it. So sometimes flowers that you may never have seen their boy parts. because generally in bouquets they're cut off because they make such a giant mess. Yeah, so they don't come back. Okay, and so they're just showing how they can cut it off, and then they can use a little paintbrush to take the pollen from whichever flower, whichever plant they want, and put it onto the flower that they made only female. So they can totally control who's the mom, who's the dad, and where the sperm and the eggs come from. And they call that cross-pollination if you do that, or self-pollination if it's just doing itself. Okay. And so so these are some of the traits that he looked at, and so these are the ones that we'll be giving examples to. And what he did was, he had this idea that there had to be something passed in the pollen between the two, because, you knew that's how fertilization happened, and that's how you make seeds, and that there must be something that is responsible for these traits. So he's suspecting genes, right, DNA or chromosomes, that sort of thing. And so what he noticed is that when if you take one plant, like a tall pea plant, and you self-fertilize it, sometimes you get talls and some shorts, right? And if you take another tall plant and you self-fertilize it and you get tall, and you take that one and you self-fertilize and you get tall, at some point, the short never come back out, right? And they call that true breeding or pure breeding. If you self-fertilize that tall plant, you'll only always get tall. So we would now know that only the tall alleles exist in that guy, right? It's not heterozygous. It has to only have tall. Tall on tall only gives you tall if that's the only alleles that are in there. When he did the short guys, short on short always gave you short. You never got a tall out of that. So, you did it for several generations just to be sure a tall never popped back out, and then said, okay, that plant is true breeding. So we know this is homozygous short, only has the particulate matter that causes short. So that's how he could then do his experiments across a tall and a short, because he knew the tall were pure tall and the short were pure short. So he had an idea of what particles, what genes were, what alleles were in those two plants. Okay? So they kind of like a purebred dog, except, purebred dogs aren't genetically identical to each other, but maybe for one trait, all the purebreds have that same, trait. Just like the tall plants aren't identical in all their other traits, but just, he's following that one phenotype, that one gene. So that's what true breeding or a pure line means in genetics. When we talk about a hybrid, hybridizing essentially means it's not pure. It has traits from tall and short, right? When we think of a hybrid, we think of, a tiger and a lion gives you a liger, right? And again, this guy has an odd number of chromosomes, so it's sterile, can't get through meiosis, so you can't do liger and liger, right? You have to kind of like mule on mule action, right? Same idea. But when we talk about hybridization or a hybrid in genetics, we just mean tall and short, something that's heterozygous, has an allele for tall and allele for short. Okay? So a dihybrid cross is talks about two different genes that are both heterozygous. A monohybrid cross is one gene but heterozygous, a big T and a little t, right? Tall and short alleles in them. Okay. When we call something a parental generation in genetics, that would be, by definition, they're true breeding. So if I told you it's parental purple and parental white, right, we know both of these are true breeding. Okay? So, and that means, right, they have to be homozygous. And homozygous means the same. Two of the same alleles for every gene. Okay? So big P, purple, little p for white. Okay? Now, since he already did some of these experiments, he knew that true breeding purple gave purple, 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 purple. White could never give anything but that, always did white, 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 white. And when he crossed those two together, what came out in the F1, which by definition is a cross of the parentals, two true breeding together, always only gave us purple. All purple, always, only. So the white phenotype disappeared. Okay? So we know now that that means these guys were all big P, little p. They got a big P, the purple trait from whichever this mom, and the white from dad, but white is hidden, right, in the heterozygote, or masked. Whichever one shows up is the dominant trait, right? It overrides the other one. So just from this one simple experiment, we know that purple is dominant over white. Because in the F1, that's all we see. We don't see any white. Okay? And so what happened to the white? Did it go away? Did we lose it? No. Did we blend it? No. Where did it go? Yeah, it's just hiding. Okay, so again, this is the F1, this is the parental. If we cross, an F2 by definition is F1 times F1. Okay, so either a self-fertilization of one plant or any of the so it could be this guy on himself or that guy on that guy, right? That gives you the F2 by definition. So if I call it an F2, it had to be true breeding up here, had to go through this F1 hetero to this F2. And we see white coming back. Mostly purple. 3/4 of them are purple, but 1/4 of them are white. Okay? And so that's what he was trying to figure out. How does white disappear and then come back? He said it has to be some molecule that exists here, is just hiding. We just don't see it. It can't have disappeared because then we'd have to say spontaneous generation recreated alleles that, occurred in these offspring, right? Conservation of matter. We can't just have things popping in and out, right? They had to be there in the F1. We just didn't see it. So the F1 tells you what's dominant and recessive. Exactly. Whichever one you see is the dominant, or you could say, well, it shows me recessive by what I don't see. Right? So either one. So anytime you're asked something about which one's dominant or recessive, this experiment could be set up and you'd say, well, whichever phenotype shows up in the F1 is the dominant phenotype, the dominant genotype. Yes, and that by definition makes the white recessive. And so those terms, dominant and recessive, refers to who wins the phenotype. The genotype, the alleles are there, it's just do they win or not. And for any specific trait, it's either dominant or recessive. It doesn't change. So in this kind of garden pea, purple is always dominant no matter which plants you look at. That doesn't mean the purple is dominant in snapdragons or whatever, but in this type of plant, if tall is dominant, it's always dominant. Right? And so for traits, I'm not going to try to make you guys memorize which ones are dominant, which ones are recessive. Right? You'll either be told or you'll be told something about an experiment that tells you. Right? So I won't expect you to know that tall is dominant or widow's peak is dominant or tongue rolling is dominant. Right? You'll either be told, or I'll say, to true breeding, blah, 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 the F1 looked hitchhiker's thumb. Then that's dominant. Okay, so you'll either be told or, and same with the diseases we'll talk about, because I don't care about memory. Some of them you'll end up remembering just because, you'll look at them enough, but we don't need to memorize that kind of stuff. Okay. So these generally have nothing to do with fitness or natural selection, right? A trait is dominant or recessive, and that's just the way it is. If that purple trait was somehow deleterious to that plant, like bees don't like purple, they only like red, or they like white, and it doesn't, then it just disappears, right? It just goes away because they don't get pollinated, they don't make seeds, they die, right? It's not because purple was so much better than white that you get 3/4 of them, right? That's just what appears. What happens after that, if they get grazed or they don't get fertilized, whatever, that's all natural selection. But the genetics is just the genetics. It's just how it works. Okay? Okay, let's see. No blending, blah, blah, blah. We talked about genes. Let's see. Okay. So again, this is the definition of genes and allele. I talked about that during mitosis because it's a concept that seems to throw people, right? So all of us have 2 copies of every gene, right? 1 on each of the homologous chromosomes. The alleles are the versions of that gene, detached or attached, tongue roller, not tongue roller, right? It's still the gene that gives that musculature in your tongue. Okay. The principle of segregation is that the alleles come apart during meiosis, okay? That if you're heterozygous, you don't pass that heterozygous trait to your offspring, right? Because heterozygous means big R, little r. And we know if you are big R, little r, we're just gonna pretend, look, here's your chromosomes. This is chromosome 11. I don't know, I have no, I'm making it up, right? And so both sisters on that one are big R, both sisters on that one are little r, right? The sister chromatids came from replication. They're exactly the same as the originals. But this one, actually I should have drawn one blue. We'll draw this guy blue. Right, because here's grandpa, grandma, chromosomes. Okay. When you end up making gametes, what happens? That guy goes this way, meiosis 1. Right, these are little r's, these are big R's. The alleles come apart. Right, they don't stay together. You can't have big R, little r in a gamete. You have half big R and half little r. Because then if we, finish meiosis and we pull apart, oh, stop it. Oh, that's an interesting color. Right, we pull apart those guys and we pull apart these guys. Right, this is a big R, this is a big R, little r, little r. Right? Each gamete only has 1 gene, 1 of the alleles. That's all segregation is. Right? That heterozygous, those 2 alleles don't stay together into the offspring. Right? You get 1 allele from mom and 1 allele from dad. And it's 50-50 chance, right? 50% of the sperm are going to have big R, 50% of the sperm are going to have little r. Who wins the race in, the plant? Who gets down the tube fastest? Random, right? Could be the big R, could be the little r. 50-50 chance. Okay. Oh, I scribbled all over this. So when we talk about which one's dominant and which one's recessive, by convention, anything dominant is a capital letter. So that also tells you something. If I say the hairy ear gene is denoted capital R, you know it's a dominant trait. Whereas, the no-haired ear is a little r. You know it's recessive. Right? So that just automatically tells you. So if something is true breeding, dominant, it's got to have 2 big R's, right? And then if you think about it, well, what kind of gametes could this guy make? Big R's. 100% of the gametes are big R's. There's nothing else that guy could make. So if we did the plant self-fertilization, this guy would get together with that guy, we'd be back to big R, big R. That's why it's true breeding. That's all they have, right? True breeding recessive has to be lowercase, lowercase. And again, that's all they have. Take them apart, put them back together, it's still lowercase, lowercase, still recessive. True breeding. But if you cross this guy with this guy, right, and this, right, this guy's only got little r's, this guy's only got big R's, and these 2 guys get together, Now we make heterozygous. That would be our F1. What does that phenotype look like? Yeah, whatever the heck that dominant is. Whatever, hairy ears, whatever it happens to be. And then you know, right, the other one's the recessive trait. So if you weren't told which was which, and that you just had two things that were true breeding, fat and skinny, right, what comes out in the F1? Fat. Okay, that's dominant over skinny, or whatever comes out. If I said, what if skinny came out in the F1? Well, then that one would be dominant. Okay. And so that's all this little guy shows, right? A Punnett square is the cheapy way to do probability without doing math. What's better than that? We can do probability by math, and when we get more complicated stuff in 360, we have to use the math because the Punnett squares get too gigantic. But the Punnett square is just doing the probability for us. And so I don't like how they start out the little example because to me it's a little bit confusing. Because we know this guy, little r, half the gametes are little r and half the gametes are little r. Right? We know this guy, half the gametes are big R, half the gametes are big R. And so if we were going to do a representation of how you could do all of them, I prefer the Punnett square to look more like this, so it doesn't confuse people later. Right? It's just showing us the probability of all the possible combinations of these gametes. Right? So this one is a gamete, that's a gamete, this is a gamete, this is a gamete. If this one and this one got together, we'd have big R, little r. If this one and this one got together, we'd have big R, little r. If this one and this one got together, we'd have big R, little r. Oh, fascinating. It's riveting. If this one and this one got together, we'd have big R, little r. That's all the possibilities. We did every single combination. We could also have just gone big R times little r, and said, okay, this one and this one, this one and this one, this one and this one, and this one and this one. Those are all the combinations. Okay? But this stupid little square is sort of idiot-proof. You can't forget to grab one because you have to fill in the stupid square. If you end up and you have a blank square, fill it in, right? You have to be able to do that. And so this just tells us exactly what that stupid cross said, right? This is the F1, and they're all big R, little r. A cross of true breeding always gives you all heterozygous offspring. And whatever their phenotype is tells you which one's dominant. Okay? So in this case, if we didn't know anything about genotypes, we're going to cross that out. And I said a wrinkled P times a smooth P gives you all smooth P's. Which one's dominant? Smooth. Right? I didn't give you any letters, so you couldn't just go by the capitals. Or if the question says, you know, they all look smooth, which is the recessive trait? Make sure you're reading the question in case I switch it up. Your brain will want to say, think dominant, but if I'm asking recessive, make sure you go, oh, not smooth. Right? The wrinkled one is the recessive trait from that. Okay. And so then here's our little picture. What if, now this would be the F1 cross, right? Two heterozygotes, heterozygote on heterozygote is the F1 cross, to give us the F2. And remember we did the white and purple, that's when our whites showed back up, right? Our F1 guys are both smooth, we know they're heterozygous. If we set up the stupid square, again, right, half would be big R, half will be little r, right? That's what he's got to give away. Half would be big R, half is little r, that's what she's got to give away. And then it's, again, it's just over and down, over, down, over, down, over, down. And by phenotype, if we can't look at their genes and we're just visualizing, how many smooths are there? Right, there's 3 out of 4. So we can just count 1, 2, 3, and how many squares are there? 4. So 3/4. It did the math for us. Woohoo! Which we know is 75%. And then 1 out of 4, or 1/4, is wrinkled, is the recessive. If we look at the genotypes, let's say we know the genotypes, we did the sequencing, right? Then we have to consider this guy is the homozygous dominant, these two are heterozygous, this guy is homozygous recessive, right? The genotypes are 1/4, 1 and 4 are big R, 2 out of 4, big R, little r, 1 out of 4, little r, little r. Okay? These two things never, ever change. You do not even need to draw a Punnett square if I ask you, what do the offspring look like, or what are the genotypes of the offspring of a heterozygote on a heterozygote? Because you will get this every single time. You can't not. You have to. Right? What if I tell you big T, little t times big T, little t? Everybody draw this on screenshot and upload, since we haven't done anything today. Right? We set up our stupid Punnett square. It does not matter if mom, maybe it does, but not for genetics. It does not matter if mom's on top or dad's on top. Okay? And you can do it both ways and do the experiment to show yourself that it doesn't matter. It doesn't matter if we put big T, little t from this one. I'll do different colors just so it helps us follow who's who. And big T, little t down there. Because again, it's probability. We're just counting how many squares out of 4. Doesn't matter where they are. The only way you guys can mess this up is if you take a big T from one, if you put both big T's on top and both little t's on the side, then you're screwed. Right? You have to keep, the gametes from one of the knuckleheads on one side and the gametes on the other. You can do it down this way and across this way, too, if you want to just go rogue and be nutty. Okay? So that doesn't matter as long as, right, each individual, mom is on top or dad is on top, but not part of mom and part of dad, because that doesn't work. Yes, you can if you can do it quickly. Right? So fill in your square. You can tell me, so that would be a question that I might ask. I might say, what the heck am I doing? Here we go. What is the phenotypic ratio and what is the genotypic ratio? Okay, so if I say, what's the pheno ratio and what's the geno ratio? Right, the phenotype is what they look like. And so for this, since I didn't tell you what the T's stand for, you'd have to say, 3/4, right, either how many dominant, how many look recessive. And then the genotype doesn't change. You can just see that it's, the ratio for this for genotype is always 1 to 2 to 1. They have to be, right? When you do right, we'll bring our T's over first, then we'll bring our T's down, and then our T's over. Those are our genotypes. The genotypes are always just the letters, right? So if you're asked if you're given a stupid paragraph, a whole bunch of crap, and then you're asked a question, you got to figure out what of that paragraph full of crap is actually useful. And if I'm asking for genotypes, it's always going to be letters. So, homozygous dominant, what's the genotype? You'd want to pick big T, not little t, that can't be dominant. And you don't want to pick tall, because that's a phenotype. So what do we got for phenotypic ratio? Yes. And how about geno? All right, screenshot and upload. So you can do this one 1 billion times and it never changes. So that means you don't even have to do it if you just know this. This is okay to memorize, right? Anytime two heterozygous are crossed, it's 3-to-1 phenotype, 1 to 2-to-1 genotype. It's always this. It's always one homozygous dominant, one homozygous recessive, and 2 heterozygous. Yeah.
What do you mean genotype?
The genotypic ratio. There's 1 homozygous dominant.
Oh.
There's 2 of 4. Right? So this is the same as saying 3/4 and 1/4, or 1/4 to 2/4 to 1/4. Right? Because in math, if you can divide or multiply numbers as long as it's the same number, it doesn't change the value. Right? So we don't have to include the 4. So the fraction is 1/4 to 2/4 to 1/4, or 1/4 to 1/2 to 1/4. The ratio is 1 to 2 to 1. Right? The fraction is 3/4 to 1/4. The ratio is 3 to 1.
So for genome ratio, it's dominant, homo,
Yep.
hetero, and then recessive.
Yep. Exactly. Okay. So all of these monohybrid crosses have predicted outcomes in the Punnett square. You can always do the Punnett square to make sure you're not messed up, but as you move on in biology and genetics, the more that you just know what the answer is going to be, the easier it'll be for you. You don't have to spend a bunch of time. You don't want to spend time doing a Punnett square on a heterozygous cross when you want to spend more time thinking about a problem that you need more time for. Right? That one you should be able to just find the answer in 15 seconds. Right? You read it, oh, it's just hetero on hetero. She's asking how many homozygous dominant, 1 and 4. Right? I don't need to draw that to know it because it never changes. And so all of these are the same thing. Okay? And I suggest you write these out as a Punnett square to help you. We're going to do this one right now, and we're going to screenshot and upload. No, is that the one that that was the top one? Yeah. Okay, so we're going to do that cross. So you're going to do a Punnett square for me. You're going to give me the phenotypic ratio and the genotypic ratio, and then you're going to screenshot and upload. No. If there's a 0, sorry, I just changed that one. Okay. So we're going to do 2. Yeah. Quickly, because we have 2 minutes. Sorry. So if you did this guy, right, the next guy is that one. So you're going to do 2 squares, 2 uploads. We're going to do that one? Yes, double. Ooh, double. I'm hungry. And then hetero on recessive homo. I guess I could have drawn them on the same page for you guys. That would have been helpful. The other one is this one. Page 2. Sorry. And then after you get your answer, you're going to go back a couple slides and look and see, right, yes, I just proved what Malone said that always happens. And so again, for this test, for you guys, you'll have time to do the Punnett squares for these. In genetics, you don't get time to do anything monohybrid. You have to just know. But for these, you can just, you'll have time. But if you save time on these by knowing it, it'll give you more time on harder stuff that, you know, that you're allocated 1 minute, you get to use 2 minutes or 3 minutes. Yeah, you can do it. Well, let's do 2 uploads. Because, you know, we need points. Right, don't we? Yeah. Yeah, so we'll use all 3 today. Woohoo! Okay. So in this case, right, but we would actually write this as a 1, right? Because it's 4 out of 4, and 4 out of 4, so we call it 1.
And then, so that it,
Yep,
okay.
exactly. It would be 1 to 1, because you could do, because like 2 out of 4 to 2 out of 4 is, yeah, so you can do least common